present to you our seminar speaker. Well, actually, this is part of the biochemistry 8530 course. This is our last lecture. And this is our, well, visiting lecturer in the course, and also Rice invited speaker. He's going to be participating in the Rice retreat this weekend. I'm very pleased to present to you Dr. Jeremy Boss. Dr. Boss got his bachelor's at State University of New York at Albany, as well as his master's in biology and his PhD in biology. This was done under the mentorship of Dr. Richard Sittemer. Then he did a postdoc at Harvard University Department of Biochemistry and Molecular Biology under the mentorship of Dr. Jack Strominger. Then he accepted a position right after he got his finished his postdoctoral training at the Emory University School of Medicine, Department of Microbiology and Immunology, where he's been since then, and rose through the ranks up to full professor, which he achieved in 1997. And his research areas of interest are molecular mechanisms of regulation of immune system genes, regulation of the human MHC complex class 2 genes, regulation of genes by TNF, and MTA3 and BCL6. So I'll let you have the process.
Thank you, Carmen, for the invitation, and to everybody for welcoming me here. And I look forward to the retreat and going kayaking and whatever we do. So, I prepared a talk that covers, well, the title, Spatial Organization and Epigenetic Regulation of MHC Class 2 Genes. And it's split up into about 3 different parts. And the first part will talk about chromatin structure around the gene, around the genes themselves. And after the introduction, you'll see where I'm going. And then how the locus is arranged and how these novel elements seem to influence how the entire locus controls expression of these genes. And then at the end, it just turns out that there happened to be a cell line that was sitting in my freezer for 15 years that had an interesting phenotype that somebody had sent to me and said, well, we can't figure this out, Jerry. Maybe someday you will. And so we pulled this, we had some ideas, and we pulled the cell line out, and I'll describe to you sort of a dysregulation of this system that was found in a patient with acute lymphoblastic leukemia. And these are this patient's cells that had a very odd phenotype with respect to this system. So that's sort of how we're going to do this. I'll be careful of the time and watch it. If you get real tired, just wave and maybe we'll take a break if you want. And if you have any questions, since it's a class, you should really interrupt me and we can get a dialogue going. And this is for you guys. So, all right. So what you're supposed to see here is a rainbow. And Emory University is it has a lot of its endowment in money because of Coca-Cola, right? So Coca-Cola made a major donation to Emory University. And what you're supposed to see here is the rainbow that indicates where the secret formula for, you still can't see it, it's too bright, for the Coca-Cola formula is to make Coca-Cola. You know how Pepsi would pay a lot of money for that. All right, so that joke didn't work because you can't see the rainbow. Or it's just a bad joke. Either way, so let's go to...
That's what the pot of gold is?
Yes. And, this was working a few seconds ago. There we go. So we're going to talk about B cells. It's actually, all of this stuff is in the human system. And so a B cell starts off as a hematopoietic, coming from a hematopoietic stem cell as a pro-B cell. As a pre-B cell, it rearranges the immunoglobulin genes, and you start to see heavy chain on the surface, and you get a full immunoglobulin IgM form on the surface that can recognize antigen. And this immature form leaves the bone marrow, and it develops into a mature form in which you're expressing 2 types of immunoglobulin on the surface. And this is the B cell that circulates through your lymph nodes and your spleen and will respond to antigens and protect you from viral and bacterial challenges. But the way the B cell gets activated is it has a molecule called a major histocompatibility complex, class 2 molecule here on its surface, and that will present antigens. You'll see a picture of that in a second. once this B cell gets activated, some of them become plasma cells, which produce high levels of antibody. And so what I have focused on for actually quite a long period of time is understanding the regulation of this system, of these molecules. You can see the developmental progression of these molecules, and then when it transforms into a plasma cell, they lose the expression of these genes. And so we're going to be talking mostly about how the genes get turned on. All right, so here's, have you all taken immunology at all? A little bit. So what you're seeing in this rendition here is basically 1/4 of an immunology course summarized to one slide. So basically, here's a pathogen, gets taken up by a macrophage or a dendritic cell, and the pathogen gets degraded, and the peptides go into these lysosomal compartments where they come in contact with one of those MHC class II molecules. And the peptides that are generated from the pathogen get loaded onto the MHC class II and transported to the surface, where the peptide and MHC2 complex on the surface, right, can then be recognized by a class of T cells called helper T cells, CD4 cells. The recognition is through a T cell receptor. It's very specific. And when this recognition happens, the T cells become activated. They divide many times. And some of them become memory cells. Others will just produce cytokines and help regulate the immune system. Still others, while in the lymph node or spleen, will seek out the B cell that has on its surface the antibody to the pathogen. And so in this way, you're looking for the B cell that has the antibody molecule that will basically block the pathogen from infecting or becoming dangerous. This same interaction is carried out through an MHC class II molecule and peptide and recognized by the T cell receptor. When this happens, there is an amplification of the B cells. You get proliferation, and again, many B cells, and ultimately forming plasma cells. But this whole process is controlled by the expression of MHC class II molecules. And if there's no expression of MHC class II, this process stops. And there's a disease called the bare lymphocyte syndrome, in which the patients don't make these, they don't transcribe MHC class II genes, and so they fail to do this. And so they are very susceptible to any type of infection. And it's actually, they ultimately succumb to infection unless they get a transplant. Okay. So starting in the genetics of the MHC locus, this is on chromosome 6 in humans. And you can see that there's a lot of genes. Each of these symbols represents a different set of genes. For the MHC class II, each of those molecules that holds a peptide is made up of an alpha chain and a beta chain. And so DRA can go with DRB1, DQA with DQB1, DPA with DP1, DPB1. And there are other molecules that help put those peptides inside an MHC class II molecule, such as the DNA, DMB, the DOB, and the DOA, and all of these sort of are co-regulated. So you have a stretch of about 650,000 base pairs of DNA, which has 14 to 15 genes that are responsible for presenting antigens, and they're co-regulated. But the genes that are shown in this color blue here, TAP1, TAP2, PSMB8, and PSMB9, are not co-regulated. So they're regulated separately. They're expressed all the time, whereas the rest of these genes are expressed only in B cells, only in macrophages, or dendritic cells. And they can all be induced by, in other cell types, by interferon gamma. Okay? So this is the system. So we've got, in B cells, a constitutive expression of all of these genes, but in epithelial cells, all of them are off except for these guys. And if you treat the epithelial cells with interferon, all of these genes come on. We know from the bare lymphocyte syndrome patients that they have, the patients fall into 4 complementation groups. Right? So that means 4 different genes. And one of the genes that's defective in the patients is a gene for this molecule called class II transactivator, or C2TA. And the other 3 groups of patients are defective in a DNA binding protein subunit, and these are the 3 different subunits. So we spent many years trying to figure this out, and we found that, in fact, it turns out that a lot of these patients are being discovered, and people have been sending them to our lab over the last couple of months to try to figure out which of the genes the patients are defective in. It's been kind of interesting. Okay, so how do these things work? So that's sort of the animation that there are, actually it was much faster than I wanted. All right. So there are 3 cis elements that sit in front of all the MHC class II gene products. They bind, now let's see if it works, RFX, CREB, and FY. And then these 3 proteins serve as a scaffold for this non-DNA binding protein called CPTA. So each of the patients might be defective in one of these 3 RFX subunits or in CPTA. Right? So the genetic basis of the disease is a transcription factor. Right? These 2 sets of factors are always there. And so this also took a long time to work out, many years. And then we started asking the questions, is this all there is? Right? And so that question comes up a lot. And the answer was, well, when we figured all this out, it was in the absence of thinking about chromatin and how chromatin might affect gene expression. And so, as you know from your course, to fit all the DNA inside of the nucleus of a cell, it has to be wrapped up and packaged into chromatin. And the basic unit of chromatin is a nucleosome consisting of 8 histone proteins in which the DNA wraps around twice, 146 base pairs going around. And then the nucleosomes are arranged into some sort of array. And aside from this structure here, the rest of this is no one knows. Okay, so this is really imagination and cartoon artist drawing. But the idea is that if you get into situations in which the nucleosomes are very tightly packed, the DNA is inaccessible to transcription factors, and the genes are turned off, and they cannot be expressed. And so there's a dynamic relationship between gene expression down in a region that might look like this versus suppression or oppression of genes in a region that might look like this. And so you have to be able to go back and forth. And one way to do that is to modify the tails on the histones through a variety of post-translational modifications. And we began to look at some of those post-translational modifications. Let's see if that works. For one of the MHC class II genes. And those modifications are pretty much on lysine, positions of lysines. And their acetylation, right, is mostly associated with gene activation in an open complex. There are also a set of methylation marks that are either repressive or activating. And so I'm just going to show 2 sets of activating sets of experiments. Here we're just looking at acetylation on histone H3, lysine 9, lysine 18, lysine 27, or histone H4. And the overall question we were trying to ask is what are the factors that contribute to putting these modifications on? And so you can sort of visualize the modifications on the nucleosome as substrates for other proteins binding. And so you have enzymes which put the marks on. We call those writers, and writing the marks on. The enzymes or complexes that read the marks, that see them, we call readers. Okay? And then there are molecules that take the marks off or exchange them, and we call those erasers. Right? So you have readers, writers, and erasers. And together they control the chromatin environment, that opening and closing and accessibility. So we're interested in trying to figure out what some of the writers were. Okay? And so Nancy Choi, a graduate student in the lab, took the HLA-DRA locus and those XY boxes that I showed you earlier, so they're right here, which is WX, there's a W box too. So there's WXY, and then she looked 300 or 600, some interval, upstream or downstream from this spot. This is located about 150 base pairs upstream of the transcription start site. And so this little map down here gives you an idea. So here's the start of transcription. This is WXY, upstream 300, 600, 2000, and downstream elements. And looked at some of the marks. So histone H3, lysine 9 acetylation, an activation mark. And you can see it's pretty much centered on the upstream region with a peak at the WXY region. And then there's a a region up here called XL4 that also has an XY box. So it's a second XY box that sits outward, and then has a small little peak of acetylation. And the other thing to look at is the scale. And the scale will change all the time on this. But the more robust the binding, the larger the scale, okay, of course. And then we do statistics to see if anything is actually significant. And in some places, you might question, it might be statistically significant, but is it really meaningful biologically? And we can talk about that if you have any questions. But you can see here, so we have K9 marks, we have K18 marks, we have K27 marks, and we have lysine H4 lysine 8 marks. And so there seems to be a general spreading of the activation mark, suggesting that the chromatin is generally open in this region when the genes are activated. Now there are 3 different cell types. For the most part, you've been looking at the blue cell type, which is Raji. Those are B cells. They're lymphoblastoid cell line that was isolated in the 1960s from a Burkitt's lymphoma. And it was the first cell line where it was shown that Epstein-Barr virus causes Burkitt's lymphoma. Right? So EBV, and the authors of the paper are Epstein and Barr. So EBV, it's kind of amusing. So that's Raji. RG225 is a cell line that was made by a scientist named Roberto Accola in Italy, in which he was looking for genes that were regulating MHC class II. And also in the 1970s, that's when this cell line was made, he mutated Raji cells, selected for a loss of class II, and then found a cell line that was consistently deficient in MHC class II. In 1993, Victor Steinley and Bernard Mock's group discovered that C2TA was a deficiency in that cell line. And so Raji and RJ225 are very, very closely matched. They differ by C2TA and I think one other gene when you do a microarray. SJO comes from a patient with a bare lymphocyte syndrome, and they don't have the RFX proteins. So without RFX, they can't bind C2TA, so nothing should happen. And you see that there's no marks, right? And whether there's some low level of marks here in the in vitro and a patient cell line, there's nothing. And these two are MHC class II negative. Just I didn't say that. So the marks look like they're dependent upon having C2TA and everything working. And then so those are the acetylation marks. There are 3 methylation marks that surround promoters, and they're all at the same site. Lysine 4-methyl-1, lysine 4-methyl dimethyl, or trimethyl, because you can keep adding lysines, and that changes the dynamics of the chromatin. We know that methyl 1 is found in enhancers, whereas methyl 2 and methyl 3 are associated with promoter regions. And you can see here that this would suggest that this might be an enhancer region up here. A high level of ME1, very low levels of ME3. But when you get down near the promoter, okay, you can see high levels of ME3. And so the question we're going to ask is, what are the proteins that might be putting these marks on? So all of those studies are done by chromatin IP. Right? You guys know that, right? As I asked. Chromatin IP. Everybody's good on chromatin IP? Where you immunoprecipitate chromatin, and then you, with an antibody, you ask by PCR what binds, right? So when you do that, you might have a picture that looks like this, where you have cross-links holding the nucleosome or some factor to the DNA. But what about when you get into higher order complexes and you have more and more connections? You're really relying on these low level of formaldehyde cross-links to do your immunoprecipitation. And sometimes that's not good enough. And so you can add an additional cross-linker, DSG, which is shown right here, which expands the distance that you can have cross-links and gives you an additional set of cross-links. So formaldehyde is roughly 2 angstroms, but DSG has a 7.7 angstrom distance in which you can use the cross-linker. So you can cross-link more things. So this just shows that if you look at an increasing time, cross-linking with DSG and formaldehyde, you can still detect C2TA. And then at this level here, C2TA is being brought down to the promoter of the gene, but a control antibody, T-cell receptor, isn't brought down. And if you look at a coactivator, that has histone acetyltransferase activity, CBP, you can see that you basically can't detect it here, but now with the increase in the cross-linking, you can detect it. So we're building complexes that are higher order, and we have a nice low background, and we have a specific activity. So that's the control. What about some of the other factors? And so here are some factors. Here's the analysis that Nancy did for CDP. And here you're going to look at Raji and RJ225, which are the C2TA minus cells. And you can see that there's a basal line here of binding, but CDP binds mostly to the WXY box where C2TA is. The upstream region, U300, which is really very close. Okay, so it's just 150 bases up. And also to XL4, that upstream WXY element. P300 also binds here, as does GCN5, and maybe PCAF binds a little bit. You can see here. It's just, but somehow the binding here doesn't seem to be as dependent upon C2TA as CBP and P300 binding. Okay? So here are 4 different coactivators. Each of these has histone acetyltransferase activity. CBP P300 modifies K18, acetyl, K14. GCN5 does lysine 9 on histone H3, and PCAF also does lysine 14 and 18 on H3, but on histone H4 does lysine 8. So this covers all of the marks. These are probably the factors that place those marks. And you might remember I said that the system can be turned on with interferon. And so if we look in epithelial cells and we treat with interferon, we can see at no interferon levels, there's no GCN5 bound, but as we treat with interferon, we can see an increase in the binding of GCN5 and PCAF to the region. So it seems that in both systems, the interferon, I should say the interferon system works by turning on C2TA. So in the cells that don't express MHC class 2, there's no C2TA, and so they're missing one of those basic components. So, that's the slide. So what about the methyl marks? This was taken out of a paper in the Journal of Biological Chemistry, where they were looking at different types of coactivator complexes that could put on methyl marks, the K4 methyl marks. And this is also from another paper in which they isolated one of the complexes called the mixed lymphocytic leukemia complex, which puts on marks at lysine 4 of histone H3. And this is what they got from that. Look at all those proteins. There's a zillion of them. And so then we asked, are any of these or any of these associated with acetylation and the methylation, right? And so Nancy, we got antibodies to those. And so one of the complexes, Saga type, this would be lysine 9 acetylation, or ATAC type, can be differentiated by this factor here, ADA2, whether they have ADA2A or ADA2B. And here's antibodies to ADA2 and ADA2A, 2B. These aren't the greatest antibodies, but what we can see from this is that there's an increase in ADA2, suggesting that it's the ATAC complex, but not the STAGA complex. The MLL complex has a variety of proteins, MLL1, WDR5, ASH2L, and RPB5, which put the methyl marks. And if you remember, the methyl marks sort of covered a very broad area, but they're mostly concentrated at the promoter. So this might mean that if the nucleosomes and are binding the methyl marks, then the methyl transferases, that they might have the ability to, sort of reach over and methylate an adjacent nucleosome. So when we have all that together, we start to put together a model that looks something like this. So here are the DNA binding proteins coming on. They recruit C2TA. C2TA begins to recruit all these other chromatin modeling complexes, and the nucleosomes sort of change their shape and probably accessibility so that RNA polymerase can be recruited and then to transcribe the gene. Now, there's a lot of proteins here, and so I talked about, let's see, I think I had 4 or 5, around 11 chromatin modifying proteins coming on. We've got the RFXs, CREB, NFY, and C2TA, so another 9 or so. So we have about 21 proteins that are being recruited to this gene to turn transcription on. And we're just mostly playing in the upstream region that's 150 base pairs upstream of transcription. And you would think that after all this time we'd say, we figured it out and we're done. But no, there were always things that bother you when you're doing science. And you remember things from graduate school and from your postdoc. You guys will remember this too. That kind of linger, right? And so, you know, you wake up in the middle of the night sometimes sweating that you didn't understand a particular facet of the biology. And one of the things that always bothered me was that in the late 1980s, a mouse was created which had this region and a little bit more, about 1,100 nucleotides upstream. And then there was a second mouse that had about 2,500 nucleotides upstream. This mouse with 11 or 1,200 nucleotides that was created by the Matheson-Benoit groups did not express quite right, even though everything here seems to be exactly what we needed. And there had been no other biochemical evidence that we needed anything else. But when you put that gene segment into a mouse and it integrates randomly, you didn't get the type of expression you wanted. But when they used a larger construct, the expression seemed to mimic the biology. And so that said that there might be something else upstream. And well, I don't know if that interpretation is fully correct. It still sort of made us think that there were other things upstream. So what they had in that 2,400 base pair construct was another one of those WXYs, like XL4 was in the previous slide. But we started asking, were there other WXY elements in the system? So we did some in silico searches, and we found lots of these. And then there were basically, I think, 40 in total that a graduate student, Jorge Gomez, found. And he turned them X-like elements, and some of them were active. And active meant they bound RFX and C2TA, and they had histone marks. And then some of them didn't do anything. They didn't bind those factors, and we decided we'd use them as a control. So we chose one of them as our control, and that was XL9. And it was and the reason we chose it as a control was it was located in nowhere land. There was nothing here. It's between the DR cluster and the DQ cluster of genes that make up the MHC proteins. And it's 28 kb from one side, 14 kb from the other. There's nothing there. I mean, there is no microRNA, there's no other transcript. It's about as dead a region as you could want. And we thought, oh, that's great, we'll use that. And so we did. So while we were doing the experiments, Jorge noticed that if we look at just acetylation of the DRB1 gene here, you can see it's pretty nice, and the DQA gene down here. So DRB1, DQA. And then when he did the acetylation of XL9, he saw that it was higher than the two. Well, this is a region of open chromatin. And then he sort of did some fine mapping, and you know, there's lots of histone acetylation. And if he went further out, there was nothing. So there's nothing in here, and there's nothing here, just XL9. So it's in the middle of nowhere, but yet there's a lot of acetylation. And you know, we have to believe that the genome doesn't do things it doesn't want. And so we continued thinking about this, and so Paramel in the lab took XL9, and the idea was, well, if it's separating these two gene sets, the DR set and the DQ set, maybe it's like one of these elements called an insulator. Okay, and insulators have one property, in that they can prevent an enhancer from working from one side of the gene to the promoter of another. And so here's an experiment. So if you have an enhancer like SV40 and you put it in a luciferase construct, it'll drive expression of luciferase really nicely, right? You transfect it into cells. If you put an official insulator, the HS4 insulator, from the beta globin gene, between SV40 and the promoter, it's dead. Okay? If you put lambda DNA in there, it doesn't do anything. That's a good control. If you put XL9 in there, it's dead. So XL9 can prevent this enhancer from acting on this promoter. So what XL9 was an insulator. And insulators have a protein. There's one protein in mammalian cells that insulators bind, and it's called the CCTC transcription factor, or CTCF. So we have C2TA and CTCF. I'll get it right when we do it. All right? And so this just shows that CTCF binds to this segment right here, D, which is in XL9. And you can see very clean chromatin IP. So you find something like this, and you have an insulator there, and you say, okay, it's just an insulator, it's separating the genes. And you have to ask the question, does it have a biological effect? Right? And so one way to find out if things have a biological effect is to do siRNA knockdowns, right? And so you could do that. An immunologist has a tool, though. An immunologist likes to do things by flow cytometry, right? And so basically, what we did here was we took cells and we did our siRNA transfection to knock down CTCF and asked, do we lose surface HLA-DR or surface HLA-DQ from the B cells? And you can permeabilize cells and stain inside the cell as well as on the outside, so you can get both readouts. And so what you're looking at here is flow cytometry. On the y-axis is HLA-DR surface expression or HLA-DQ surface expression. And on the x-axis, you're looking at intracellular CTCF. When we look through the controls, GFP, so a non-specific siRNA, you can see you have nice HLA-DR expression on the surface and high levels of CTCF expression. Same thing with DQ. Instead of doing surface DR or DQ, you do intracellular RFX. You can see that this is fine. So the siRNA didn't perturb this. But if you look at CTCF, you can see that cells that lost CTCF expression also lost surface DR or DQ expression. So this shows that CTCF affects the expression of the MHC complex. So you have an insulator, which should be preventing the genes from crosstalking to each other, is now functioning in the expression. So this was very different than paradigm. Well, those were control genes to show that not everybody changes. So that might mean that C2TA and CTCF interact. And from my picture, the cartoon, you might have noticed that everything was interacting with C2TA. So it was a reasonable assumption that CTCF might. And so this is a pull down. And so we took Raji cell lysates and we immunoprecipitated with CTCF antibodies and came back with a C2TA Western blot. And you can see that nice strong band, control antibody nothing. This is RJ, which doesn't have C2TA, so that's a good control, blank. But you can complement, right? And so you can add C2TA back and you restore the pull down, showing specificity. You can do the experiment in the other direction by pulling down with C2TA here, IP with C2TA, and Western blot for CTCF, and you can see that it works. The epithelium bromide and DNase that's shown here, if you want to show that proteins interact with each other in the absence of DNA, you add epithelium bromide, and it intercalates in the DNA and distorts the helix, and proteins don't bind, okay? So it's a very old trick. You can also throw in DNase, of course, but the epithelium one is a lot easier. You know, everything's red, it's also toxic too. These were some other things I won't go through there. So we have a situation. We have an insulator that's binding CTCF in the middle of nowhere, and we have a promoter region proximal regulatory element that's binding C2TA, and you have to ask, do they interact? Okay? And so about 6 or 7 years ago, an assay was developed that allowed you to do that, and it's called the chromatin conformation capture, or 3C, CCC, right, assay. It's also referred to as a looping assay. But this is how it essentially works. You have two cis elements, distal and proximal, and you have lots of restriction sites in between. Like the chromatin IP, where you cross-link things, you treat cells with formaldehyde, and you capture those interactions, and you have these cross-links here. And so you can imagine that this is interacting with that through a protein. These will be cross-linked. Formaldehyde is not a great cross-linker. And so the DNA can still be cut with restriction enzymes. So you take your small amount of DNA and you put a lot of restriction enzyme in with it, and you let it go overnight. And it digests it to completion, and you end up with the DNA being cut at this site and this restriction site. You then kill the restriction enzyme by heat. You dilute the sample greatly so that you favor intramolecular interactions when you add DNA ligase back. So have any of you taken a plasmid, cut it, and diluted it, and reclosed the plasmid to remove a fragment to make a deletion? So when you do that, you dilute it out, and so it favors the ends that are connected on the plasmid to ligate to each other. Same principle here. So you generate these by adding, by cutting, and then ligating. You generate this novel ligation junction. This sequence does not exist in the genome, right? And so you can PCR amplify this, right, and assay whether or not this occurred. And if you get a PCR product, the conclusion is that this interacted with that. Okay? So this is what it really looks like. So here's the locus, DRB1, DQA1. Here's XL9. And this was the experiment. We asked, does XL9, which we call, so we have an anchor primer, and we asked, does it interact with any of these other restriction fragments? 23 restriction fragments. And that's shown down here. And you can see that it interacts with the DRB1, number 3, and the DQA1, number 22. And it's in Raji cells and not in RJ225. So the genetic control plus C2TA minus C2TA. This over here shows the fragment interacting with itself, so it actually will close back on itself. So this experiment's about 1,500 PCR assays. Just to let you know how many plates. It's not triggered. You get a model that looks like this. So in cells from the bare lymphocyte syndrome patient, there's no RFX present, so that it doesn't assemble, the factors don't assemble. In cells that are like epithelial cells that are induced by interferon to express MAC class 2, before they have interferon, there's no C2TA. This would be the same as a patient who is missing C2TA, and you still have no interaction. CTCF is always present, but when the gene becomes activated through the expression of C2TA and all the factors assembled, you get a structure that looks like this. And so now we have one gene, one insulator, one CTCF site. But I showed you at the beginning that the locus is very large. There are 14 or 15 genes. Do they all use CTCF? Do they all use XL9? Is this really what it's looking like? Or are there other ways of thinking about it? So the question is CTCF required for all MAC class 2 expression? Are there other XL9-like sites? If so, where are they, and do they function like XL9? If not, is XL9 required for MAC2 expression, functioning as a hub? Right? Are there other components associated with XL9, and can the XL9 be dysregulated? Right? So those seem like reasonable questions you would have after that paper. So here's the locus, just to remind you, here's XL9. And so to find other CTCF binding sites, we did chromatin IP chip. We basically took chromatin IP with CTCF and put it on a microarray that had just chromosome 6. At the same time we were collecting the data, 2 papers came out that were doing ChIP-seq, where they did the chromatin IP and they took all that stuff and they put it in for deep sequencing, and they found all the CTCF sites in the genome. There are about between 12,000 and 17,000 CTCF binding sites, right? So there are lots of these. We took our data, their data, combined it, and actually together we got 16 sites, including 17, including XL9. And we named them C1, 2, 3, 4, 5, 6, 7, 8, and so forth. So then the question becomes, well, you have a lot of sites. Are they all real? Right? And so to do that, to figure that out, you have to go back and do a regular chromatin, excuse me, chromatin IP, and look at the sequences. And if you put the sequences together for all these sites, then they're not very similar. The gray bars here show some of the similarity, but there's a core sequence here where they were most similar. And when you look at that core, you can generate these, the computer generates all this stuff. I mean, it used to be you did this by hand. Now you just plug it in and go. You guys are really lucky. This was horrible to do by hand. But this generates it nicely for you. It's great. So that's what the site looks like. And this turns out, from this, to match the consensus sequence that was found in those two papers, which I was really happy because, you know, I didn't see it looking at this. I would never have been able to put that together. So you can do a gel shift to look at the binding of CTCF to all the sites. And so in this case, we set it up as a competition because that's the easiest way to do it. You take a bona fide insulator site like the HS4 site that binds CTCF, you get a band here, and then you do a competition assay. You just add cold sequences, cold oligonucleotides, and you ask, do you lose the bands? And you can see C1, 2, and 3 compete nicely. C4, not so well. C6, 8, 10, and 15 don't compete. So those really aren't binding CTCF in vitro, so they're probably not real. Right? So we cross them off the list. And then we do the chromatin IP using Raji cells or RJ225 cells. And what you can see here is that, let's see, C1, C2, C3 are very nice. C4, not so good. C5, good. 6, no good. 8, no good. 10 and 11 are no good, but they bound in vitro. So in vitro they bind, but they're not binding in vivo here. 13 is not binding, and 15 is not binding. It's very consistent. So all of a sudden, the number of sites in vivo are reduced from 17 sites down to 10. So 16 by ChIP-seq technologies, and 10 left. So a little bit. So ChIP-seq isn't always right, or maybe it's not right in this cell type. Right? The caveat. If you look where they are, C1 and XL9 surround the DR subregion. XL9 and C2 surround the DQ subregion. There's a second DQ subregion. These are inactive, these are pseudogenes. They're expressed, but they make no product. C2 and C3 surround that. The DP genes are surrounded by 14 and 16. And then if you go through pretty carefully, you'll see that each of the class 2 genes is surrounded by an insulator set of CTCF binding sites. So the placement now is really very exact. And since these were generated by some duplication event, it might be that this is how the duplication happened. Of course, we don't know that for sure, but it's a nice hypothesis. So then Paramel Majumdar in the lab asked whether or not CTCF was required for the expression of all the genes, just like XL9 was responsible for DRB1 and DQA1. And so here's the experiment. You got CTCF knockdown, and this just shows the purification of those cells. And then he compares that to a DRA knockdown, which is shown here. So if you do the siRNA for DRA, it's really low. And he compares these two. And you can see that DRA, which is at the 5' end of the locus, is also affected by CTCF. And you can almost see it here. So the shaded regions show all of the genes that are knocked down. And so every one of the genes that are co-regulated by C2TA are affected by CTCF knockdown. GAPDH isn't, but these other genes that are in the locus, they're not affected. So the model hold, do they interact? How do they interact with CTCF sites? And so you do a lot of 3C. So this just shows the DR region interacting with C1. Here's C1, all the restriction sites. Here's DRA. And if this is the anchor, and you ask, do any of these fragments interact? This just shows some of the tests here, and showing that it's dependent upon formaldehyde, cross-linking, and on ligation. The two controls don't interact, so you have a nice specific interaction. So C1 interacts with DRA1. You can go across the entire locus now and put together another few thousand of these 3C assays. And then you look at the data. And the data show, let's see, For example, you already saw C1 and DRA. I think it'll be another one. And here, just to show you DPD1 interacting with C14. And you'll note that C2, and it just works its way across, that if you take one of these sites, it'll interact with either of the neighboring genes. Okay, you can go across. And so the genes that are next to CTCF sites interact with them. Again, it was done in Raji and RJ225, so you can see the dependence on CTTA. But you'll notice that there are two, this interaction here and this interaction here, that appear to be CTTA independent. And I'm going to come back to that in a minute. So if we ask, does C1 interact with DRA and so forth down in this direction, who does DRA interact with? We ask the question from the other side. Oh, this just shows they're inducible by interferon. And so ask me the question, who does DRA interact with? So here are all the CTCF sites. And so DRA is interacting with C1, right, here. It's also interacting a little bit with XL9. This is roughly 150,000 bases away. It doesn't seem to interact with the rest. I did do this. Okay, so DRB1 can choose XL9 or C1. Maybe it interacts with C2, but it really doesn't like it very much. So there's some preference going on. Okay, I'll do one more. Okay, DQA1 likes XL9 or C2, so it goes here, here. It doesn't seem to go here, and it doesn't like any of these others. And so as you go down the locus this way, you can see that the genes are interacting almost in register with the closest set of CTCF binding sites. And they like some, they don't like others. But it might just be a distance thing, and it might be a proximity or a stochastic. So in other words, it can interact if it's not occupied. Okay? So that would be one way of thinking about it. If you remember a few seconds ago, I told you that there were some interactions that appeared to be not dependent upon C2TA. So, for example, I think I was telling you that DPA1 had problems interacting with C16. Well, it interacted with C16 in both RJ and Raji. And that's because the restriction fragment had another insulator on it. And so what we were seeing in that assay wasn't a CTTA-dependent interaction. It was a CTCF site interaction. So then we asked the question, so that said maybe these sites interact with each other. See how it's getting complicated? You have the promoter region factors interacting with the insulator, but you also might have insulators interacting with each other. Okay? And so that's shown here. If we look at each of the insulators and the CTCF sites, and we say, well, who do they interact with? And the experiment set up is to look at each of the insulator segments again. And remembering, here's the map up here. And so C1 interacts with itself. That's shown here for Raji and RJ225. So that's the gray bars, the self interaction. But then it interacts with XL9 and C2, a little bit with C3, and then it kind of dies off. XL9 interacts with itself. It will interact with C1 and with C2, a little bit with C3, and it falls off. C2 has a broader set of interactions. It'll interact all the way up here and a little bit downstream, but not in this part of the locus. C3 is the most prolific, and that will interact with both sides. But you can see the register moving down. And it seems that the boundaries are about 250,000 bases in which these insulators will interact with each other. Okay. So, obviously you have to ask questions about what's required. And so if you do the CTCF knockdown, you can ask, is CTCF required for these interactions? And so to make sure you've knocked it down, you look at chromatin IP. And this just shows that if you knock down CTCF, you lose its binding. So that's just a nice control. It's a lot of assays, but it's the control, the right control. And you can ask, well, if you knock it down, do you lose the interactions between DRA1 and C1? And that's shown here. This is the wild type, and here's the knockdown. Here's the control that didn't bind. Again, XL9 interacting with DRB1 and the knockdown. Here's C2 interacting with DQD1 or DQA2, and again, the knockdown removes the interaction. The same thing happens for the CTCF sites, in that we ask C1 interacting with itself. That is shown here, and then we just have the other interactions going across the locus. The cell fragment you always see, but when you remove CTCF, you remove the other interactions. Okay? So that's the transcription factors. Right? So the question we're not answering is whether or not the site itself is required for transcription. Right? So you know, a geneticist would have you knock out the site, right? Or you knock out the cis element. Those are the oldest experiments we could do. But this is a human cell, and so you can't really just go in there and knock out the site. It would just take years of breeding. You know, you just can't do it. Grants don't go that long. And it's not. I'm sorry, I'm on television. It's not ethical either. So to do those experiments, and you also want to be able to, and these are large pieces of DNA, right? And so the distances are 20, 30 kilobases. And you can't do this with normal plasmids, but you can do it with bacterial artificial chromosomes. And so these plasmids, right? This one's about 200,000 base pairs. So it's a big plasmid. And this particular one contains DRB1, XL9, the entire DQ cluster, C2, and just misses DQA2. So it's got 3 genes and 2 insulators. And so if you transfect that into cells, and so this is a human clone, we can transfect it into mouse cells, mouse B cells, and ask, do we see MHC class 2, human MHC class 2? And the answer is yes. So that means we can go in and start to manipulate it. And manipulating the bacterial artificial chromosomes is called back recombinering. So like engineering and recombination. So it's done by recombination. You know, just like you would make a targeted mutation in a mouse, you make a vector that has homology arms, right? Some selectable marker, right? And then another set of homology arms. And you take the back and you place it into E. coli, and you put the targeting vector into the shuttle vector into E. coli, and through homologous recombination with one end, you don't need both sides, it basically integrates the entire shuttle vector. And then you can select against one of the 2 markers in the plasmid itself, the shuttle vector. And so the bacteria can't grow unless it deletes out the plasmid. And so then it does a second recombination event. So you have a targeting event, integration, and you have a resolution. So you put your DNA in, and then you resolve it out. And so you can basically generate deletions and insertions within this large plasmid. And it takes about 3 weeks to do each one. And it's remarkably efficient. The people who worked with DNA viruses, like these herpes viruses, have been doing this for years. They just didn't tell anybody about it for a long time. And so basically, here's the wild type. We made a deletion of XL9, a deletion of C2, and then we made a back that had a double deletion, right? And then we asked, what happens? And so this is the expression pattern that you get. So I think in the end, you'll see that everything's not quite black and white. But what you'll see is that here's the wild type, and you look at the expression of DRB1, so it's this gene on the wild type back, and it expresses really nicely. And you can see here it's 150-fold. Okay? If you delete XL9, expression goes down to about 10-fold. Okay? If you delete C2, it's still doing pretty well. So it doesn't use C2. If XL9 is present, it really likes XL9, and it's not real crazy about C2 because you have a 10-fold or so reduction in expression. You knock out both, and the expression goes really low. So that tells us 2 things. It tells us that there's a preference, but it also tells us that there are no other insulators, right? And so that's required one of these two elements, XL9 or C2, are required. If you look at DQA1, expression is pretty low. And DQA1 is not expressed very highly anyway in these cells at the endogenous locus. But it's sort of XL9 deletion. It's still expressing a little bit. C2 deletion, maybe a little bit of expression, maybe not, probably pretty low. You knock them both off and you're really down at the baseline here. DQB1 has just the opposite effect of DRB1. And you can see you knock down a C2, brings expression down very low, whereas a deletion of XL9 expression is pretty close to wild type. And so just going back to here, DRB1 is close to XL9, it prefers that. Even in the back, which is a limited number of choices, it prefers XL9. DQ can use either XL9, DQA can use either XL9 or C2. DQB prefers C2 over XL9. And so basically these sites are required, and there appears to be some preference that we didn't know about. We can also do 3C on the transfected bags. We can do chromatin IPs, and we can do everything on this, except the numbers are really low. So your background has to be really even lower when we do these. But basically, got some circles. Here we go. So some of these are controls. If you look at the interaction between XL9 and DRB1, if you remove XL9, you lose the interaction. But if you delete C2, you can still see the interaction between XL9 and DRB1. So this goes really well with the RNA data, right? So that makes sense. But this one didn't go as well. So if you look at C2 and DRB1, there is an interaction with C2. It's lower than the interaction with XL9. But if you, of course, if you delete C2, it's still there. The opposite is the same for DQA. So it looks like in the back they can interact with the different sites by 3C, but the interactions aren't productive, right? So this interaction, if I go back a slide, right, is not very productive. Because you have a lower level expression. But perhaps this level, considering the scale, is the same as DQ. And so what you're really seeing is it interacts, but it's not an efficient interaction. And so DRB1 chooses the best interacting insulator to interact with, which is XL9 in this case. So that's what we think. Okay. So answering the question, are there other factors and things involved? It turned out that last year, I guess it's actually now 2008, a number of groups found that doing ChIP-seq, that this protein called Cohesion was found associated with about 75% of CTCF sites. So Cohesion, or cohesins, are proteins that hold cystochromatids together in mitosis. And so you know when you got the chromosomes replicating and they go to metaphase plate, they pair up. And so cohesin is required for that pairing. And then when cohesin becomes phosphorylated, it releases it and the chromosomes can separate to the daughter cells. Okay? And so people thought for a very long time that cohesin was only involved in that aspect of cell biology. But then people found that it was expressed during other parts of the cell cycle. And so, you know, and these guys were looking to see where it was. They found it at the CTCF sites by ChIP-seq. And if they knock down cohesin in a reporter assay like the one I showed for CTCF, the ability of the CTCF's insulator site to block enhancer activity was reduced. So cohesin appeared to be involved in enhancer blocking activity. So we asked whether it was bound to the CTCF sites, and I know you don't quite remember that picture earlier of the ChIP assay for CTCF sites, but this is almost, we could just change the names. It's essentially identical to the CTCF sites. And I don't have any data to show you on this, but if you knock down the cohesin subunits, you basically lose expression of DRA. It goes down. It's not destroyed. DRA, DRB, DQ. So cohesin is involved. And cohesin's got 4 proteins, and it forms a circle, it's thought, to help link these things together. And so we come up with a model, and this was the first model that I was sort of drawing, was that if you had lots of these CTCF sites, you get this kind of cute little structure, right, that goes like that. And that seemed, at the beginning, before we had much of this data, to explain things. And then I thought, well, okay, you can have that, but why not that, right? And I don't think this is true either, because we don't see the interactions between this locus and this locus down here. And so we had something that became sort of more complex and not as freehand as the other thing, where we have CTCF sites interacting with each other, we have cohesion going around them, and we have all the genes, right? And I've just drawn one configuration. And this is in the off state. In the on state, the promoters of these genes come down and they interact with these foci here and here. And so this is one configuration. And you can ask, and we don't know the answer to this, but just to think about the arrangement. And so some people kind of think that these CTCF sites form these long loops. And what's inside the loop can be expressed. And what's outside the loop isn't expressed. And so if you had typically if you had an enhancer inside the loop and a gene down here, the enhancer somehow can't interact with the gene. And that would be the picture that you would get for an enhancer blocking activity. So maybe that only the genes inside of the loop can be expressed, and in this cell that would have this conformation, the DQ genes can't be expressed because they're really not inside of a loop. They're sort of in an interloop place. Whereas a DQ2 and DQ2 genes are within another loop, and they can be expressed if they come down and interact. So that was one conformation. But the data showed several. C1 can interact with C2. XL9 can also interact with C3. If you do that, you sort of kind of get this crazier loop. You've got these four genes being expressed, and these guys are also in a loop. And so this type of conformation might allow all of these genes to be expressed. And maybe the DO gene here can't interact, and so in those cells, it would not be expressed. So unfortunately, we don't have the technology to answer that question of how they exist right now. But one experiment we can do is ask, do these types of structures occur in a cell, and if they do, is there any property that's associated with them? And I think this is the only dark slide that we have. I don't know if you guys can see this or not. Can we turn off the lights? Yeah. So this is not 3D fish. It's just regular fish. And what you're looking at are RNA transcripts. So it's RNA fish and immunohistochemistry. And so what you're seeing here is the DRD1 locus being expressed. Both chromosomes are being expressed. And here's Pol2, polymerase 2. And you can see when that there's a dot here. And so what you see when you look at Pol2 is there's lots of speckles. And this is the activated form of Pol2. And so this is where transcription is occurring in the nucleus. And you can see that this locus sits right on top of this dot here when you do the merge, here and here. In RJ225, there's no expression, so that's just a control. And here, looking at the DQ genes, there's a dot here and one here, and you can see the overlap. So these dots are often called transcription factories, where you have an accumulation of the products necessary for transcription, RNA splicing, and things like that. You can turn the lights on. So we think that the association of these loops act to bring everything together to places where all the factors are there to modify the chromatin, to initiate transcription and move things forward. So the last part, how are we doing? Are we doing good? I'm going to end early, so it won't be. So the last part is about this cell line that I said at the beginning that this guy Jerry Nepom, who's in Seattle, sent me in the late 1990s and said, well, we've gotten this far on this, but we haven't gotten any further. Maybe you guys can figure it out at some point. So it sat in the freezer for years. So, 15 years. And what was special? So the Lazarus was the guy's name who was the author of the paper. And they isolated from a patient, this is 1978, and this is when people were isolating cells and trying to grow them in culture. And so at the Dana-Farber, a laboratory isolated LAS221 from a patient and grew this acute lymphoblastic leukemia cell line in culture, and in fact, just growing the cell line was a pain. Okay, that's how things were back then. They also isolated from the paper a normal cell that they transformed with Epstein-Barr virus, right? And so they had a normal B cell, and they had this abnormal cell that at the beginning they called a null phenotype. They really didn't know what it was. And ultimately it turned out to be a pre-B cell leukemia, okay? And if you look at, and so what Jerry Nepom's group did was they looked by flow cytometry, this is our data, but they basically found that HLA-DR was expressed in these cells, in both cell types, in both cells. And, but HLA-DQ was expressed in the so-called wild-type EBV transformed B cell, but not in LAS221. And so you had a dysregulation of expression. So we looked at the RNA for DRB1, DQA1, DQB1. You can see here that DRB1 is expressed in both cell lines, but the DQ genes are not. And so when I saw, so I thought, oh, maybe it's XL9, right? Because I was very XL9 centric at the time. And so I was thinking, oh, that would be a great hypothesis. What if the leukemic line got a mutation in XL9 and deleted it? And then you would lose expression from the DQ genes, but because DRB1 can use C1 and DQ really didn't, that would explain everything. So we cloned out the XL9 regions and sequenced them. They were wild type. We cloned out the promoters and sequenced them. They were wild type. It was really very disappointing. So let's see. So then we asked, about, so this is, well, does CTCF bind? So once we knew the sequence was there, I thought, well, maybe it doesn't bind, right? I'm still sticking to my hypothesis. I am not letting it go. And you can see in LAS338, the wild type, it's binding, but in the cancer cell, the tumor cell line, it doesn't bind to XL9. These are nice controls. And so that was great. And so then it would be even better if CTTA and RFX bound to the promoters of the genes, but not XL9. That's where my hypothesis starts to fall apart. And so we looked at RFX5 and C2TA, and you can see that they both bind to the DR in both cell lines, so the DRB1. So this is the one that's being expressed. But if you look at RFX5, doesn't bind in LAS221, and C2TA doesn't bind in LAS221. So there's a fundamental difference now across the locus. CTCF site isn't binding its guys. The promoters aren't binding the factors. Okay. So then maybe it's something else. And so CTCF is known to be involved in imprinting. And the classic example for imprinting is the H19IG F2 locus in the mouse and in human. And there's an imprinting control region where CTCF binds. And the way imprinting is regulated in that system is by DNA methylation of the imprinting control region. So a methylated region of CTCF binding site won't bind CTCF. So then we ask the question, is this region methylated? CPG methylation, right? And so CPG methylation is associated with closed chromatin structure, inaccessibility to restriction enzymes. And so this assay is called a methyl dip assay. And so it's like a chromatin IP, but it's much easier to do. You don't have to cross-link, you just take cells, you isolate DNA, you sonicate it so it's small and reasonable, you boil it, boiling's good, kills everything except RNAs, and then you do an immune precipitation with an antibody to 5-methyl cytosine. And so that pulls down the cytosines that are methylated, the CPGs that are methylated. And then you have the IP, you can do PCR. And so here's PCR across the locus. And so we'll start at this side because it's easier to explain. You have the 2 control fragments, C2A, C2B, and A. These are a couple of kilobases away. You've got the C2 site, and it comes down with the methyl dip assay. The DQB1 promoter is highly methylated in the pre-B cell leukemia. DQA is highly methylated. XL9 is really highly methylated, but not in the wild type. But then, you also have CPGs that are in non-expressed regions that are methylated, and that's pretty standard across the genome. But you get to this spot here, and these guys are methylated. But by the time you get to DRB1, it's no longer methylated. So methylation, DNA methylation, looks like it's the cause. So somehow this region got methylated. And we would propose that XL9 got methylated. You lost CTCF binding and it destabilized the region. Right? So, but the methylation is just in here. It's between the insulators. So another property of the insulators, and I didn't tell you earlier, was that some of them have what's called barrier activity. So they function as barriers. Well, barriers to what? Barriers to heterochromatin encroachment. And so the idea is if you have DNA methylation in heterochromatin, it can only go as far as the insulator, and it stops. It doesn't go past that. And that's seen in everything that has like variegation. So like if you're looking at the plants, right? This is the easiest way to describe it. And you see flower petals that are white and red, right? Everybody goes, oh, that's variegated. What that means is the reason that you have the different colors is because heterochromatin came in and shut down the gene that was making the red, right? And so like in a fly, sometimes there are flies with red eyes, and then there's a gene called the white gene, which is actually red eye color. But if they integrate the white gene in different places in the fly, you can sometimes see a speckle pattern, and that's because the gene is sometimes on or off. And that's due to heterochromatin formation and encroachment. But if you protect the white gene by barriers or insulators, in a fly it's called gypsy, it's always red. And so that's what you're seeing here. Perhaps these are now functioning as a barrier element. So, can we reverse this? And the answer is going to be yes, you'll see. So there's a drug called 5'-deoxyazacitidine, which blocks DNA methyltransferase activity, right? And so it blocks an enzyme called DNA methyltransferase 1, DNMT1. And you treat cells with 5-deoxyazacitidine. It's 5-deoxyazacitidine. It's very labile, so you have to treat over and over and over again. But now if you look at the chip, you can see that here we're untreated and treated. We look to C2TA, and these are in LAS 221 cells. So, at DRB1, you can see that it really didn't change the binding. But at DQA1, you can now see C2TA bound. You can see RFX bound at DQA1 here. CTCF now is bound to some extent at XL9. So by treating with 5-deoxyazacitidine, we remove DNA methylation, and we can also now regain some of our 3C loops between XL9 and DRB1. So that loop now appears nicely. You can see here in the wild type it's there. In LAS 221, we don't see this loop. So that meant that DRB1 is using perhaps C1 over here on this side, right? DQA1, again the same thing, but 5-deoxyazacitidine treatment, we now restore the ability to form these loops. And then you say, well, do you get expression? And of course, I wouldn't drag you through all of this without saying, yes, we do. This is what you saw at the beginning. This is the treated, untreated, and it's plotted badly. I apologize for this, but this is over the untreated. And so DRB1 actually goes up, maybe fourfold with treatment in LAS 221, maybe because it can now use XL9 and C1. But DQ goes up, 40-fold, basically the limit of our detection, when we treat with 5-deoxyazacitidine. So by removing the DNA methylation, we reverse the phenotype of this tumor cell. So this is the summary of what I've told you. And so basically, here's the locus. I've placed in all of the CTCF sites in here. So MHC class 2 genes require the action of an insulator and the insulator factor CTCF to drive their expression. It functions through interactions between the proximal promoter regions. There appear in the model systems to be preferences, such that in the case of DRB1, we're choosing XL9 over C2, and even though in the model system it can interact with C2,
this interaction isn't as productive. And it might be that C2 is not a very efficient insulator. And so the DQ genes can either use XL9 or C2, but maybe DRB1 has a higher affinity for the transcription factors, and it competes out, and there's a competition. And so the DQ genes, which are not as highly expressed, lose out. It turns out that CTCF has 11 zinc fingers in its structure. Zinc fingers combine to DNA sequences. They recognize 2 to 4 nucleotides. And from my slide all the way at the beginning, we had about a 13 base pair consensus sequence. So if you need 3, right, we have plenty of zinc fingers. We're only using 4 or 5 out of the 11. And so maybe what you're doing with some of these is you select different zinc fingers, and the affinities and interactions between CTCF and these factors down here don't work as well. It's just a hypothesis. We can actually test that one. And each of the CTCF sites interact with each other. And so you've got this organizational structure that we would propose that, along with Cohesion, to stabilize the complexes, that we're forming some type of transcription factory or transcription location in the nucleus, which allows for a higher rate or more efficient transcription. I really like the idea that the CTCF sites demarcate the different sets of genes, and to me that sort of may explain how the system becomes duplicated. Or that to be efficiently duplicated for an evolutionary advantage, you have to take a CTCF site with it. That's it. We know that, I didn't tell you this, I didn't show you the slide, but if you knock down CTCF, you lose some of those histone marks in the promoters. So the CTCF interaction is required for efficient histone acetylation and methylation. Okay. And then lastly, the LAS experiments, something that's sitting in your freezer for 15 years that you remember. This is where the only thing that the mentor and the PI can do is have that 15-year memory, because there's nobody in the lab that was there when we got the cells. And the amazing part was we were able to find them, right? I mean, no one knows what happens to the freezers. So the people who did the work, Caramel Majumdar did a lot of the, he did all of the 3C work that you saw in the last stuff. And he was helped by some of the people in our lab. This is Halloween. Here's Caramel. And here's Nancy as a pirate. And here's me as a hobbit. And we had a pumpkin, I think, Mickey Mouse, and Irish. I don't know what. Well, I don't know what it is. Actually, I can't even tell who that is. Anyway, the official acknowledgement slide has the people in the laboratory who contributed to this. Here again, Caramel and Nancy. Joshua and Royce made the constructs for the backs, and Jorge basically made the initial discovery of CTCF interactions in this thesis work. And I didn't talk about any of these others. We're helped by a variety of collaborators. Brian Chadwick did the RNA fish experiment. We have a variety of collaborators with Rafi Ahmed and Jim Riley and Paul Wade, and we're supported by NIH. So thank you guys. Are there any questions?
So I do have one question. So basically, this region, when it's inactive, it's to say that it's methylated or hypermethylated normally, and then those methylations have to be moved so that the CTCF can bind and NFY,
all those guys.
And so that we could think maybe on three events happening. That first, the methylations have to be lost. That will be the first event. The second event, all these factors enter, except for C2TA, which will have to enter after induction, maybe by interferon or other, and that will be the third event. But then, how accessible is that chromatin in the methylated stage, and
right, so
thinking who might enter and
who's first?
Exactly.
Yeah, so I think the the causal events of much of epigenetics are still unknown, right? And so in development, the DQ genes are expressed later than the DR genes, okay? When one thinks about how do you and so if the pre-B cell line, I sort of painted it, the picture that I painted was that this was a cause or a part of the transformation mechanism. But what if this was an early B cell where perhaps DQ wasn't expressed, and so the locus is normally methylated early, and then somehow it's released from methylation. So how do you lose methylation is one question. And so so this has been a battle that's been going on for 30 years, loss of DNA methylation. So it's been known for a long time that on early embryogenesis you lose methylation, there's a wave, and then you regain it. No one really knows the mechanism. And so there's passive and active. Passive would be, well, you just replicate the DNA, and one strand is methylated, the new strand isn't, and you just fail to put on the methyl marks. So that would be passive. So as you go through cell divisions, you lose methylations. Very easy to imagine. Then everybody else wants to have some enzymatic function that removes the methyl group. All right. So lots of enzymes that are proposed, all have failed any rigorous test. Currently, there are two systems that are thought to remove methylation. So there was one proposal that the AID, the activation-induced cytosine deaminase that's involved in somatic hypermutation and class switch is involved in DNA methylation removal because it mutates the Cs, and then they're replaced. Now, I haven't seen that grab on much, all right? And the second is that the Cs can be replaced, the methyl Cs can be converted to 5-hydroxymethylcytosine, which is not recognized by the methyltransferases. And so then following a round of division, they're gone completely, never remethylated, okay? Now, whether there's something that removes 5-hydroxymethylcytosine or not, I don't know. That's not known. But the 5-hydroxymethyl story is gaining some ground, all right? So let's say that there's an active mechanism, all right? And I actually think there is, because there's many cases in the immune system in which it is removed. But to direct something, you have to have a transcription factor, right? To direct all of this, there has to be some DNA sequence recognition. Otherwise, things wouldn't work the way you want, right? We end up as blobs. Since we don't want to be that, we want to have all the different organs, there must be some mechanism. So there has to be sequences that are accessible. And so the enhancer regions are often open chromatin. And so if you can bind the factor at an enhancer region, then that allows that factor to recruit coactivators and start maybe stochastically modifying the chromatin and recruiting other proteins until it hits a spot that has another factor, and you can form these loops. Right? And so I think mechanistically, one can imagine many ways in which a factor has to have accessibility. And then whether or not you remove the methylation passively through replication, right, which is easy to do, think about, or you actively remove it through enzymatic activity to allow the next step, that's still up in the air. For us, I think finding the methylation pattern in that pre-B cell leukemia was very lucky, right? And lucky that the person sent me the cell line too, because it could still be sitting in Seattle and no one would ever work on it. What happens next with it, I don't know. What we'd like to know is about the preferences of these sites. We'd like to know what's happening in the mouse. We'd like to know how the sites are working. And so we're constructing 22 of these bags that ask questions about, is there a preference for sites? If you move them around, put them in a different place, do they still work? And to understand how this is a very novel role for CTCF. Usually it's just thought of holding things together in a loop, and that's about it. Questions? Tired you guys out. Okay, clear.